Hi everybody, and welcome to our first discussion of exercise physiology, covering Chapter 1, the structure and function of exercising muscle. To start our discussion, we need to discuss muscle tissue. There are three distinct types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is probably what you're the most familiar with. This is connected to bones via tendons and assists with locomotion and other physiological processes. Our skeletal muscle is under voluntary control, which means I choose when it contracts, and it is also multinucleated. This means that each cell has multiple nuclei, so we can produce high amounts of protein during protein synthesis. Skeletal muscle is also highly organized and striated, which gives it a striped appearance. Our second type of muscle is also striated, but also branched. This is cardiac muscle. Cardiac muscle is found almost exclusively in the heart, and it only has one nucleus. This allows it to have coordinated contraction. Our cardiac muscle is also under involuntary control, which means I do not decide when cardiac muscle contracts. Instead, this is regulated in the brain by higher order commands. Our third and final type of muscle is our smooth muscle. This is typically found in hollow organs, blood vessels, and other locations around the body. It is also under involuntary control and tends to have spindle-shaped cells which allow for highly coordinated, smooth muscle contractions. We will now dig a little bit deeper to discuss the skeletal muscle anatomy. The entire muscle is surrounded by something called the epimysium, which is a thick binding layer that is going to hold the individual fasciculi or muscle bundles together. These individual fasciculi are going to be surrounded by something called the perimysium. This is what allows our individual muscle cells and muscle fibers to be differentiated. Our individual muscle fibers will then be surrounded by the endomysium, and this will be made of myofibrils divided into individual sarcomeres. Our sarcomeres are going to be the actual working parts of the muscle. We also have something called the plasma lamina, which is the cell membrane, which will fuse with the tendon. This is what's going to allow an action potential to travel down the muscle and eventually create contraction. Our plasma lamina also maintains the pH and transports nutrients. Also within the skeletal muscle are satellite cells. These are important for muscle growth and development. They also help aid response to injury, immobilization, and during training. Additionally, we have something called the sarcoplasm. This is going to be the specialized cytoplasm of our muscle cells. It is going to have several unique features such as glycogen storage and myoglobin. Also within the cell are going to be T-tubules, also known as transverse tubules. These are going to serve as extensions of the plasma lamina. These are going to also assist in carrying action potentials deep within the muscle fiber. Our sarcoplasmic reticulum, which is also part of this system, is going to aid in calcium storage. Now we are going to look closer at the myofibrils and the sarcomere anatomy. You find the myofibrils by going from the muscle to the fasciculi to the muscle fiber to the individual myofibril. There are going to be hundreds to thousands of myofibrils per individual muscle fiber. Within these myofibrils, we will find our sarcomeres. Our sarcomeres are going to be our basic contractile elements in our skeletal muscle. These are going to lie end to end for the full length of the myofibril. Our myofibrils have a very distinctive striped appearance, which gives us our striations. These are going to be made up of A bands, which are our dark stripes, our I bands, which are our light stripes, and then two distinct zones. We'll have the H zone, which is in the middle of the A band, and the M line, which is in the middle of the H zone. These are going to each individually be separated by Z discs. These are our common boundary structures between the sarcomeres. When it comes to muscular contraction, there are two distinct filaments that come into play. Actin, which is known as your thin filament, and is going to be found in the I band, and myosin, which is your thick filament, which is going to be found in the A band and the H zone. Myosin is going to be made of two intertwined filaments with globular heads. These globular heads will protrude 360 degrees around the thick filament axis. These are going to interact with our actin filaments to form contraction, Within our myosin, we are also going to have a molecule named titin, which will act as our stabilizer. Actin itself is composed of 3 distinct proteins. Actin also has a myosin binding site. Within this, you're going to also have tropomyosin, which will cover the active site of actin at rest, and troponin, which will be anchored to the actin, which will move tropomyosin. Both of these structures are going to be anchored at the Z disc. This will also equally be spaced out by titin. So finally, titin is going to act like a spring. This stiffness will increase as muscular activation and force development also increases. Our titin is going to expand from the Z disc to the M band. When calcium binds to titin, increasing muscle force will occur when stretched. Titin is mostly addressed in something called the winding filament theory, which is outside of the extent of this course. You just need to know that titin is a thing, and that its main job is to stabilize sarcomeres and center myosin. Titin also prevents overstretching. So, when we get to contraction, this is going to be controlled by something called a motor unit. This is going to be a single alpha motor neuron and all of the fibers that it innervates. More operating motor units means there's going to be more contractile force. Within this motor unit is going to be something called your neuromuscular junction. This is going to be the synapse between the alpha motor neuron and the particular muscle fiber. This will be the site of communication between the neuron and the muscle. So, how does a contraction actually take place? We use this initial start of the muscle fiber contraction we refer to as excitation-contraction coupling. Your first step is that you will receive an action potential that starts in the brain. So some sort of stimulus has told the brain, hey, I need to contract this muscle. This action potential is then going to travel down and arrive at an axon terminal and release a substance called acetylcholine. This is a particular neurotransmitter that is going to stimulate muscular contraction. Acetylcholine will cross the synapse, bind to an acetylcholine receptor on the plasma lamina. From here, this action potential will travel down the plasma lamina to the T-tubules. Once it reaches the T-tubules, this is going to trigger a release of calcium ions from the sarcoplasmic reticulum. These calcium ions will enable actin-myosin contraction and start the sliding filament theory, which we'll discuss in a minute. This is going to be a visual representation of excitation-contraction coupling. So, in summary, we're going to have a response to an action potential. The motor neuron will release acetylcholine, which is going to cross the synaptic cleft and bind to the receptors on the plasma lamina. If enough acetylcholine binds, an action potential will be generated in the particular muscle fiber. This action potential will then trigger the release of calcium ions from the terminal cisternae of the sarcoplasmic reticulum into the sarcoplasm. The calcium ions will then bind to troponin on the actin filament, and the troponin pulls the tropomyosin off of the active sites, which allows myosin heads to attach to the actin filament. Looking deeper into this, we will discuss the sliding filament theory. Once the action potential arrives at the sarcoplasmic reticulum from the T-tubule, the sarcoplasmic reticulum will be sensitive to that electrical charge. This will cause a mass release of calcium into the sarcoplasm. From here, calcium will bind to troponin on the thin filament. At rest, tropomyosin will cover the myosin binding site, which blocks actin and myosin interaction. The troponin-calcium complex will then move tropomyosin out of the way. Myosin will then be able to bind to actin, and contraction can then occur. In the relaxed state, there is no actin-myosin interaction at the binding site, and our myofilaments overlap a little bit. In the contracted state, myosin heads pull actin towards the sarcomere center during the power stroke. Filaments will slide past each other, and the sarcomeres, myofibrils, and muscle fiber will all shorten. After the power stroke ends, myosin will detach from the active site. The myosin head will then rotate back to its original position and attach to another active site further down the myofibril. This process will continue until a few different things occur. First, if the Z disc reaches the myosin filaments, contraction will no longer occur. Or if the action potential stops and calcium leaves the sarcoplasmic reticulum. To summarize, in the ready state, the myosin crossbridge is tightly bound at a 45-degree angle to the actin filament. ATP will then bind to the myosin, allowing it to release from the actin filament. ATPase on the myosin will hydrolyze the ATP to excess energy, and the myosin head moves away from the actin filament. ADP and a phosphate group will remain bound to myosin. The myosin head then moves 90 degrees and binds to the new actin molecule. The myosin head releases phosphates, which will then initiate the power stroke, where it tilts back to 45 degrees, pulling the thin filament towards the center of the sarcomere. After the power stroke, the myosin head will release ADP and return it to the ready state. This process continues until the ends of the myosin filaments reach the disease discs, or calcium is pumped back to the sarcoplasmic reticulum. So, how do we actually develop the energy for muscle contraction and relaxation? The most important molecule for this is ATP, or adenosine triphosphate. Adenosine triphosphate, or ATP, will bind to the myosin head. ATPase on the myosin head will turn into ADP and a phosphate group plus energy. Once an action potential ends, electrical stimulation to the sarcoplasmic reticulum stops. If we want the muscle to relax, we have to wait for calcium ions to be pumped back into the sarcoplasmic reticulum, where it can be stored until the next action potential arrives. This process of pumping calcium back to the sarcoplasmic reticulum requires energy, or ATP. Without calcium ions, troponin and tropomyosin will return to the resting conformation, which covers our myosin binding sites. This prevents actin-myosin cross-bridging. Each individual muscle fiber has a unique characteristic. We tend to refer to these as muscle fiber types. The three that we will be concerned with in this class are type 1, type 2A, and type 2X. Type 1 fibers consist of about 50% of all the fibers in the average muscle. They have peak tension at about 110 milliseconds, so we refer to them as our slow twitch fibers. Our type 2 fibers have peak tension at about 50 milliseconds, therefore we refer to them as our fast twitch fibers. Type 2A makes up about 25% of the fibers in the average muscle, and type 2X makes up about the remaining 25% of fibers in the average muscle. So, how do we actually figure out the muscle fiber types? We have a couple different ways to do this. The first is we can look at the variable speed of the myosin ATPase in each of these muscles. Fast myosin ATPase is typically used for fast contraction cycling, so we tend to see this in type 2 muscle fibers. Our slower myosin ATPase causes slower contraction cycling, so we tend to see this in our type 1 fibers. Another way we can do this is to perform a muscle biopsy. This is an example of the picture shown here. To do this, an individual will have a small, somewhere between 10 and 100 gram piece of muscle removed from a particular area. This is usually done during a punch biopsy. That sample is then frozen, typically in liquid nitrogen, and then sliced and examined after being stained through a microscope. Another version we can use is gel electrophoresis. Type 1 and type 2 fibers are going to have different types of myosin. We can then use gel electrophoresis, which you may be familiar with for processing DNA, and separate the different types of myosin by their particular size. We can also look at the sarcoplasmic reticulum of each of these muscle fibers to type them. Type 2 fibers tend to have more highly developed sarcoplasmic reticulum. This is because the calcium ions will be released faster. Therefore, we have about 3 to 5 times faster force development. We can also look at our motor units. Our type 1 motor units tend to have smaller neurons of less than 300 fibers. Our type 2 motor units tend to have larger neurons, which are greater than 300 fibers. This table has a summary of the different characteristics of the muscle fiber types. I would highly suggest pausing the video here and taking some notes. This is an additional table which helps with the structural and functional characteristics of the muscle fiber types. This would be another great opportunity to pause the video and take some notes. Looking forward, our muscle fibers will have different types of force production. Our type 2X muscle fibers will produce more force than our type 2A muscle fibers, which will produce more force than our type 1 muscle fibers. This is going to be due to various factors like the structure of the sarcoplasmic reticulum, the number of motor units, and so on. Looking at this, all of our muscle fibers reach peak power at about 20% of their peak force. So they can produce more force, but not necessarily more power after 20%. Each person is also going to have a unique ratio of type 1, type 2A, and type 2X muscle fibers. Our arm and leg ratios tend to be similar in the same person. So if somebody tends to have higher amounts of type 2X muscle fibers in their legs, that tends to follow in the arms as well. Our type 1 muscle fibers will also be predominant in our endurance athletes, and our type 2 will be predominant in more of our power athletes. There are certain muscles in the body, though, that tend to be strictly type 1 or type 2. For example, our soleus, depending on who you're talking to, one of our calf muscles, tends to be type 1 exclusively in everybody. Looking more specifically at these fibers, let's look at type 1 fibers during exercise. As I've stated before, our type 1 fibers tend to possess more aerobic endurance. This means that they can maintain exercise for prolonged periods of time, but they will also require high amounts of oxygen for ATP production. This means that we can recruit them for low-intensity aerobic exercise and activities of daily living. They will also very efficiently produce ATP from fat and carbohydrate sources. Type 2 muscle fibers, on the other hand, in general, tend to fatigue much more quickly, which means they have poor aerobic endurance, but they also produce ATP anaerobically and much faster. Type 2A muscle fibers will produce more force but fatigue faster than type 1, so we tend to use these more for short endurance activities, something like a mile run, maybe an 800. Type 2X, on the other hand, are seldom used for everyday activities and tend to be found in short explosive movements like sprints, such as the 100-meter dash. So what determines our muscle fiber type? So first and foremost, we have genetics to consider. This is going to help determine where and what alpha motor neurons are going to innervate our particular fibers. And it's also going to contribute to how our fibers differentiate on that particular alpha motor neuron. We cannot change our genetics, so there's just kind of some factor of muscle fiber typing that we can't change. However, there are also training factors that affect muscle fiber type. When you train, you start to differentiate your endurance training, strength training, and detraining, and this will impact the ability for our muscle fibers to do work for us during exercise or physical activity. Our training can induce very small changes within our muscle fiber type. So we cannot go from a type 1 muscle fiber to a type 2 muscle fiber. However, I can take that type 1 muscle fiber and make it act more explosively. Or I can take that type 2 muscle fiber and make it act more aerobically. I'm not changing from a type 1 to a type 2 or a type 2 to a type 1, but I am adapting it through training to characteristics that are more like a type 1 or type 2 muscle fiber. And then finally, the last thing that will determine our muscle fiber type is aging. We start to lose our type 2 muscle motor units as we age. Therefore, you tend to become less explosive and you tend to not have as much sprint endurance. In terms of muscle fiber recruitment, this is going to have a couple different methods for how we do this. The first is, how do we alter our force production? If we want to produce less force, we have to utilize fewer or smaller motor units. When we want to produce more force, we start to bring in our larger or more motor units. Our type 1 motor units tend to be smaller than type 2 motor units, which means that when I recruit muscles, I tend to go type 1, type 2A, and then type 2X. Also, when we go to recruit these, we use something called the size principle. Whenever we participate in physical activity, we want to recruit the minimum number of motor units required for that particular movement. So first, we will recruit our smallest type 1 motor units. Then we will go to our mid-sized type 2A motor units. And then we will go to our largest type 2X motor units. And we will always recruit in the same order each time. This is called the size principle. So the order of recruitment is going to relate directly to the size of the alpha motor neuron. So, can we use fiber types to determine athletic success? We do know that type 1 muscle fibers tend to be predominant in endurance athletes, and we know that type 2 muscle fibers tend to predominate in sprinters and other explosive athletes. However, success in athletics is also affected by other things, like your cardiovascular function and conditioning, an individual's motivation, their training habits, and their muscle size. So while muscle fiber type does help give us some insight into somebody's potential for athletic success, there are a lot of other factors that come into play. Next, we need to talk about not just the fiber type, but how we ask that muscle to contract. There are two main types of muscle contractions. We have static contractions and dynamic contractions. Static contractions are when a muscle produces force but does not change length. That means our joint angle does not change. We have myosin cross bridges that form and recycle, but they are not actually sliding past each other. On the other hand, we have dynamic movement. This is when a muscle is going to produce force and there will be a change in length. That means we're going to have a joint movement produced. We have two different types of dynamic muscle contractions. We have concentric, which is as the muscle produces force, it is actively getting shorter. This is the most familiar type of muscle contraction. This means the sarcomere will shorten and the filaments slide towards the center. On the other hand, eccentric muscle contraction is when the muscle produces force but lengthens. This means cross bridges form, but the sarcomere actually lengthens. An example of this is when you go to lower a heavy weight. Additionally, when we go to generate force, we have to go in a particular order of motor unit recruitment, the size principle. So our type 2 motor units tend to produce more force than our type 1 motor units, which produce less force. So fewer small muscle fibers versus more large muscle fibers kind of determines how much force we need to produce. If I need to continually produce more and more muscle force, I'm going to have to do this with a couple different things. We are going to impact the frequency of stimulation or the rate coding to produce the required force in that particular muscle. When I have just one stimulus come through, this is going to be a twitch. All right, one stimulus, one small force production. When I start to get 3 or more stimuli coming together, then we're going to produce something called a summation. I've had multiple stimuli that create multiple force contractions. If we continue to build up this stimulus, we're going to create something called tinnitus. So this is a continual stimulation of the muscle or a forced contraction. Now that we've discussed the size principle of motor recruitment and how our muscles are actually stimulated to contract, we need to talk about two important relationships with muscle contraction. And the first is the length-tension relationship. The length-tension relationship states that there is an optimal sarcomere length to produce optimal overlap, which will produce optimal force development. If our sarcomere is too stretched or it's too short, little or no force will be developed. You can think of this about trying to do a bicep curl. If your arm is fully stretched and that bicep is fully stretched, it can be hard to get the weight going. Also, when you get to the very top of your contraction in your bicep curl, it can be difficult to finish the rep. This is because in both these situations, you are outside of your optimal length-tension relationship. The final relationship we need to discuss is the force-velocity relationship, or the speed-force relationship. This states that with concentric muscle contractions, maximal force development will decrease at higher speeds. On the eccentric side of this, maximal force development increases at higher speeds. This means that there is an optimal speed to produce the most force. I want you to try something. Make a fist with your hand, and I want you to try to do bicep curls as quickly as you can. You can probably do a lot very quickly before you get fatigued. However, if I were to ask you to do the same thing, but put a 50-pound dumbbell in your hand, could you do the same amount of reps? Probably not. You could probably maybe do one very strong, struggled bicep curl with a 50-pound dumbbell. whereas you can do many with no dumbbell. So, something to think about. Thank you all so much for listening, and I look forward to seeing you guys back for Chapter 2.